Okay, everyone, here we are again. You at home watching this, me, your favorite online lab instructor. I know I am because I happen to be your only online lab instructor, but that's okay. We are here in the A&P lab for the last new information for your lab semester. So this will round out our lab experience with lab number 9. So this one covers the senses. We say special and cutaneous, even though we're mostly focusing on two special senses, the eye and the ear. Not really that much with the cutaneous senses. You'll learn about all that in your lectures, knock on whiteboard. We hope sometime just before or shortly after you see this online lab. So what we have today is pretty straightforward stuff. First, we have the eye anatomy from models, the dissection of a sheep eyeball that I will take you through, then histology, a microscope slide of an eye. As you probably are already aware, I always prefer to go models first, then the dissection, then the histology. Remember this for the future. For many of you, please don't weep at home. This will probably be your last online lab with me. Who knows what the future holds for us? Who knows if the college wants to continue using my online labs? Who knows if we'll be here in person, hopefully, next semester, probably with masks on, but then you can get your hands on the models. You can get squirted by eyeball juice, aka aqueous humor, like anybody. So hopefully, this will make some sense to you in this online digital format. We also have ear anatomy from the models and an ear microscope slide. Then we have how to test for. Now remember, how we perform these tests is what I can ask you. For localizing a stimulus, color blindness, and an astigmatism. And I'll take you through those, not very complicated. The big chunk of today's lab is the eye and the ear anatomy and histology. I've got a bunch of different eye models, so let's start out by taking a look at them. Ba-dum-bum. Hey, what do you think you're looking at? Gotta have my fun in lab where I can find it. So here we have a couple eyeball models. I have a bunch of eye models. You can see some of them arrayed out here. Many of them are just copies of the same thing over and over again. But we'll take a look at these, and I can tell that this eyeball is a left one, and this is a right one. Not that that particularly matters for us, but I can tell that by the location of the lacrimal glands that I see right here. You'll probably hear about those a little in lecture, producing tears for you as you weep over the end of this online lab, of course. Now, what we're going to do is go through your lab manual and look at some of these structures on my various eye models, but we won't go in order. What we're going to do is look at some of the external structures first before I start dismantling these eyeball models. And yes, I understand it creeps you out a little bit, these two eyes just staring into your soul like this, and that's the reason I set up the shot. Anyway, so hopefully the audio quality will be good in this video. I'm still fighting the loud noise of the air handlers. If the audio quality is bad, I think I will fire the audio engineer, who is the guy speaking right now, and he'll lose his salary. Oh, I don't pay him. So I guess that's too bad for our audio engineer. So again, hopefully the audio quality will be good enough for us to get through this. Let's begin in your lab manual with what are called the extrinsic eye muscles. I can see these on several different models Of course, you can see these model representations of skeletal muscles on and around these eyeballs. I'll start with this one. And what we have are a set of skeletal muscles attached mostly to the sclera, the white of our eye, which allows our eye to move around, up, down, look at different things. So as we look at this list, you will see 1, 2, 3, 4, 5, 6 extrinsic eye muscles. You see 4 of them that are called rectus muscles. Rectus, of course, referring to very straight. Now, if I look at this particular model, here's a helpful hint for us to orient ourselves to start out looking at these rectus muscles, and that again is this lacrimal gland right here. I choose this model because I can see where the nasal bone is, the nose would be right here. This is this person's right eye as they look straight at you. That makes this the lateral side where the lacrimal gland is located. And if you don't mind a little bit of alliteration, lateral, lacrimal, lacrimal, lateral. It's on the lateral side of this eyeball. So if I rotate it, you see this rectus muscle, which is located on the same side as the lacrimal gland. This one is the lateral rectus muscle. Lateral rectus muscle, right there. I'm going to start with that one because I have a good anatomical landmark. I can also see above it, and I'll zoom in just a tiny bit for us. I can see above this 2 muscles here. See the division? This is 2 muscles. The lower of these 2 is the superior rectus muscle. Look for that name. Lateral rectus, right here where the lacrimal gland is. Superior rectus, right there. What's this muscle located above the superior rectus? This is the most superior of our eye muscles that you must name. This one is called the levator palpebrae superioris. I'm going to say that again. The levator palpebrae superioris. This is the one that controls your upper eyelid. Let me rotate this around. Lift it up a little bit to get a good angle. So this very, very top muscle, the levator palpebrae superioris, would be attached to the skin of your eyelid, which would be right here, to open your eye. So the first 3 we see, lateral rectus, superior rectus, levator palpebrae superioris. That's how you say that one. But there are still more muscles, aren't there? So this eye comes apart nicely for us. Watch me remove these top 2 muscles. I'll remove the lateral rectus because we still have 2 more rectus muscles to look at. And they're located right here and right there on the other side of the optic nerve, everyone. This one at the bottom is called the inferior rectus. This one on the inside, the nose side, in here, let me try to give you several views. I take out part of the eyeball. This one right here is the medial rectus. Inferior rectus, medial rectus. Now that still gives me two more muscles to know the names of, the obliques. Let's take a look at those. So I've reassembled this eye model without the eyelid on it. So I can see again lateral rectus with no lacrimal gland. I could use the nasal bone because this would be the medial side, this would be the lateral side. Lateral rectus, I'll remove it. Superior rectus, the bottom one, right here. Levator palpebrae superioris. I'm going to pop that off again. And now, with the help of a little bit of camera work, I can also see medial rectus, inferior rectus, those are pretty easy. 4 rectus muscles, lateral, superior, inferior, medial. But what's this one? Hugging the upper inside of the eye socket. This muscle right here is the superior oblique muscle. The superior oblique muscle. Look for that name in your manual, and let me show you exactly how to know this muscle when you see it. I'm going to do an extreme close-up on my eye model. Notice right here, right there, there's a tendon for this muscle. Sorry. There's a tendon for this muscle that shoots through this little loop like a pulley. This thing is called the trochlea. It's not on your list. This muscle is controlled by the trochlear nerve. That's the superior oblique muscle, which makes this essentially 90-degree turn and attaches to the white, the sclera of your eye, from above at an angle. Superior oblique muscle. If I look down, I can see this muscle sweeping across the bottom side of the eye at an angle, the inferior oblique muscle. Let's look at these on a similar model. So notice our lacrimal gland. That immediately orients me, right? Lateral rectus, superior rectus, levator palpebrae superioris. I can see right here the inferior rectus. If I do a little model dissection, right here, the superior oblique muscle, whose tendon, its insertion goes, they always have it cut so I can remove the eyeball, goes right through this trochlea right up here. So the trochlea, the superior oblique muscle goes through it. Down at the bottom, inferior oblique muscle. If we look at this eye model right here, there's nothing wrong with it, but it doesn't show every single muscle. So I have a rectus muscle there, a rectus muscle there, down at the bottom, and then what do I have? Oh no, levator palpebrae superioris on this one. And while I can see some oblique fibers, it's kind of tough to tell who is what. And I have no nose, no lacrimal gland to orient me, do I? Let's look at the eye muscles on this thing. Since I do have a lacrimal gland, I can see lateral rectus. I can see my 2 up at the top. Let me zoom in for you. Superior rectus, levator palpebrae superioris. Inferior rectus, inferior oblique. Here's more inferior oblique right there. I can see the tendon coming in for the superior oblique muscle. Which is located right here, superior oblique. And what doesn't show up very well on this model is a medial rectus, unless I turn it just so you can see it right there. Let's do the muscles on one more model. So look at this one, lacrimal gland, lateral rectus, superior rectus, inferior rectus. Remember, the rectus muscles have straight fibers. Superior rectus, levator palpebrae superioris, medial rectus, and if I start removing some structures here, I can see down at the bottom, notice the fibers running this way, the inferior oblique. What does this muscle not have? A superior oblique. So remember, not every model is perfect, but you must have something that will orient you for those rectus muscles, these extrinsic eye muscles. Lacrimal gland. If there's no lacrimal gland, you would need a nasal bone. Lateral lacrimal. Extrinsic eye muscles. Make sure you know those. Okay, let's continue with some of these external eye structures, things that we can see on the outside. So, first, let's just address this lacrimal issue. So we have lacrimal glands on the lateral side of each eye. But we have other lacrimal structures on your list you're responsible for naming. Lacrimal canals, lacrimal sac, nasolacrimal duct. So, first off, this is the lacrimal gland. Right there, we already mentioned it. That's on the lateral side. On the medial side, next to the nasal bone, we see this structure, which is the lacrimal sac. And notice right here, lacrimal canals. That's what those are called. Lacrimal sac, lacrimal canals. If I use the other side, the right side eyeball, so lacrimal gland, pretty easy to find, lateral side, then lacrimal canals, right here and here, and lacrimal sac. If I keep looking downward, I can see, I can see the lacrimal sac becoming what's called the nasolacrimal duct, right there. So lacrimal sac is the bulb on the top, nasolacrimal duct is below that. On this model, I have a great lacrimal sac, but I don't know if you can notice, it just stops there then. So I don't have a nasolacrimal duct, which would be below that. Lacrimal gland, lacrimal sac, lacrimal canals, nasolacrimal duct down there, which is the best representation I have of it right there. Notice lacrimal sac right up there and lacrimal canals. We also have the palpebrae, which are your eyelids, best represented here, here, and here on this model. Upper, lower eyelid, the palpebrae. Don't have a lot of model representation of that, but we have it right here. Also listed as an external structure is the orbit of your eye. Remember we talked about that before, way back in lab number 3. Seems like a long time ago, lab number 3, doesn't it? The orbit is the bony ring that goes all the way around here. Remember there's a supraorbital and an infraorbital margin. This model only shows me the lower part of the orbit. If I look at this big and somewhat creepy model right here, I can see more of the orbit. Now do not confuse the orbit with the socket. The socket of the eye is this depression that the eyeball fits in. The orbit is simply the ring that goes all the way around. So the orbit of the eye, the bony ring that surrounds it, also as an external feature, we would have the sclera, or the white of the eye. Remember, that's what these extrinsic eye muscles attach to, the sclera, the white, the outer fibrous tunic, white because of all the collagen there. And continuous with that is this really piece of connective tissue with a thin sheet of epithelium on the anterior side, thin sheet on the posterior side. This is the cornea. This is the thing upon which you contact lens wearers put your contact lens. I don't really understand how you do that, it just creeps me out. But I know some of you just do it like nothing. You just throw a contact lens right on the cornea. It's slightly different type of collagen, smaller fibers with some proteoglycan gel in there, so it stays nice and clear while we're alive. So the sclera is the white of the eye, the cornea is the front clear portion of the eye. And if we look straight into the eye, I can also see externally the pupil, which is the hole, and the iris, which is the colored part. The pupil, which is the hole, and the iris, which is the colored part of the eye. And I'm going to throw this one out at you. Your eyes are brown, no matter what color they are, unless you suffer from albinism. Our eyes are brown based on the color of melanin. Ooh, ask your lecture professor about that. It has to do with reflection of light rays of various wavelengths. Getting all physicsy on you here in our last lab. But the iris is the colored part of the eye. The pupil, actually a hole. If I remove this cornea, which I should be able to pop this right out. There it goes. I think you can tell that this pupil is actually a hole. It's not an object, it's a hole. Iris, pupil, sclera, cornea. Come over to this larger model, palpebrae. Let's remove that. sclera, cornea, they only gave me half of it here. I have another half, it's right here. Cornea, iris, pupil, part of the orbit. I can see, again, these are all external structures of your eye. Only two structures left that I would say are external structures of the eyeball. The first one, and it is on our list, I just missed it the first time I looked, the trochlea, this small pulley of connective tissue I see right there. The other model I have that shows it, right here, look at that, it loops around this tendon of the superior oblique muscle. That thing is called the trochlea. Now, as we go through these models, and you look at this and the practice section, remember the same rule applies in this last lab as applies for every other one. If I show it to you here in the instructional video, and I show it to you in the practice video, either place, that makes the model fair game for your quiz or practical. Remember that. But I don't have that many real variations of eyeball models here. The one last thing to show you, I suppose, is the easiest. I say it's the easiest because we've seen it before. Orbicularis oculi muscle. The orbicularis oculi. These are a lot of the structures that are external to your eye. The other structures, we're going to have to start dismantling the eyeball to see those, but we will. Now let's talk about some of these internal eye structures. So I want you to follow along on the list as I dismantle a couple of these things. Now, not going to go exactly in order, but we will hit everything. That's the plan. So we see the sclera right here and the cornea for each of these eyeball models. And I need to start taking a few things apart so I can see a little bit farther into the eyeball. Now, I will start with this one right here for a particular reason. This is the one that has half of a cornea for us. So if I tilt the camera up and we look down at it, you can see this space right here, which is part of what's called the anterior cavity of the eye. Our eye has two cavities in it, an anterior and a posterior. Now watch what I do with this model, if it'll work out for me. So I take the top half of the sclera off. This dark material here is the choroid coat. I pop that off. Then replace everything right here. I'm going to turn this thing sideways. I'm going to raise the camera and angle it down a little bit for us. So what I want you to notice is this structure right here. It's removable. This is the lens of the eye. And when I see that lens, that separates. It's the divider between the anterior and posterior cavities. The anterior cavity is everything in front of the lens, or from the lens to the cornea. This area right here would be filled with that thin watery fluid you will hear about in your lecture called aqueous humor. We will also see it in our eyeball dissection. I don't have a model of it because it's a thin watery fluid. but it would be in front of or anterior to the lens. So aqueous humor would be right here where I'm wagging my pointer around. This big plastic bulb that you see right here, this is representing for us the vitreous humor, which is located behind or posterior to the lens. This is the posterior cavity. So the posterior cavity is occupied by this vitreous humor. In front of the lens, the anterior cavity occupied by aqueous humor. The aqueous humor cycles around, in a fashion similar maybe to cerebrospinal fluid. You heard of that. The vitreous humor, that's essentially permanent. It just sits there and gives a lot of the eyeball its shape. Much thicker, gloopier fluid you will see when I do the eyeball dissection. So I'll always have a model representation of vitreous humor, never of aqueous. Now I'm going to try to do this as well as I can with a little super close-up right here. And I want you to look at your lab manual. Under the term anterior cavity, you see that divided into two sections. So remember, anterior cavity is everything from the lens forward. But see this wall right here? That's the iris, everybody, the colored part of the eye. And that separates the two chambers of the anterior cavity. So watch what I do here with my probe. From the iris to the cornea, anterior chamber. From the iris to the lens, right here, let me readjust my model of my lens. From the iris to the lens, much smaller posterior chamber, right there. Posterior chamber, anterior chamber of the anterior cavity. Now, I know the question as I wag my pointer around at you. I know the question you'll ask, how do I indicate that on an online lab quiz? Well, a single dot right here in front of the iris behind the cornea, the most specific name you can pick is anterior chamber. You would have to pick that one. Same thing if I put a dot right here between the lens and the iris right there, posterior chamber. If I somehow indicate the area totally in front of the lens from here to here, anterior cavity, and the area behind the lens, posterior cavity. Let's look at the same structures on a different model right here. So if I take off this rectus muscle, some sclera, remember there's dark things called the choroid. Here's the cornea, and where the tip of my probe is going right here between the iris and the cornea, that's the anterior chamber, isn't it? I'll just pull this cornea out, and I will separate this eyeball. So notice the lens right here, and this is the representation of vitreous humor. This is the representation of vitreous humor right here. And I want you to notice something else I can see here on the vitreous humor. Look very closely. Look deep into my eyes. See these lines here? These stripes that you see in the vitreous humor? I'll remove the lens. These things right here, these are suspensory ligaments that actually connect muscles to that lens of your eye as it sits there. So look for all these stripy structures. Those are suspensory ligaments. Over here, I can see the same things. Notice all those suspensory ligaments. These are actually attaching musculature to your lens to change its shape and size. So I'll remove the lens from this eyeball. I'm going to pop out the vitreous humor. And I can see this tan nervous structure of the eye, the innermost layer or coat. This is the retina that I'm tapping with my finger right here. I can see all three layers on this eye model right here. Sclera, choroid, the dark one, and it's actually very, very black when you actually see it, and then retina. And that vitreous humor glob sits right there and helps hold the retina against the back of the eyeball. The retina is where our photoreceptors are. It's essentially the screen of your eyeball, so light comes through the pupil, through the lens, focused on the retina. We need to change the size and shape of this lens to focus on objects that are different distances away from us. And we have to use muscles to make that happen. So I want you to watch what I'm going to do here. I'm going to rotate this all the way around. Do you see these white stripes right here? Those are also representing these suspensory ligaments which pull and release to change the shape of this lens for focusing on objects that are near or far. Notice on the other side, just external here to those suspensory ligaments, this structure right here is typically called the ciliary body, sometimes called the ciliary muscles. Look for that in your list of structures. The ciliary body right here behind the iris, remember the colored part, attached to the suspensory ligaments, pulling when contracting these smooth muscle cells out on the eye. So we have the ciliary body, stripes, suspensory ligaments, and the lens. Here coats, we have the retina, the dark choroid, and the white sclera. Looking at this eyeball model, iris, pupil, choroid layer, choroid coat, lens, which I just popped out, suspensory ligaments right here. Let me remove the vitreous humor, and you can see this retina right there. Think of this view, you've probably seen it in various movies when they want to show you very high-tech biometric readers. You know, they have somebody look at an optical reader and they get a picture of their retina with all the blood vessels. So this is the nervous part of the eye we'll come back to in just a minute. What I want to look at first is the back side of this. So again, see my whitish, tannish suspensory ligaments attached to the ciliary body or ciliary muscles right there. And just for lecture, it will be right here where your pupillary constrictors and dilator muscles would be that you probably hear about in your lecture. So suspensory ligaments, ciliary body, right there. Now let's get back to our retina because we have a couple interesting structures that I can see right here on this model very, very nicely. Not every model shows this. First, I want you to look at this white circle you see right here. That is called the optic disc. I'm going to say that again, the optic disc. It is where all the axons of all these neurons gather up and shoot through a hole, your optic foramen, sort of like a rag being sucked down a drain. And right behind it, that's where the optic nerve comes out. The optic disc is a section of your retina that has no photoreceptors on it. So sometimes this is called your blind spot. And it's almost always shown to us as a white or a very light colored circle. The other circle that we see right here, this one much smaller, that's called the macula. Sometimes called the macula lutea, because it has it has a little yellow dot in the middle. That yellow dot in the middle is called the central fovea, or the fovea centralis. So look for this in your lab manual. Macula right there, and fovea centralis, Latinized, right there. Don't confuse that with the optic disc. Some people call this the blind spot. This thing, the macula, I call this the sweet spot. What do I mean by that? Well, the sweet spot is the place we want the image to land when we look at it. Imagine this eyeball, if I was to put it back together quickly, like so, like this. Don't we constantly move our eyes around looking at things? Because the light rays that shoot through our pupil, where do we want them to land? We want them to land right on that macula, because that's where we have a lot of our cones. Those photoreceptors we call the cones, which give us high definition or high acuity or high resolution vision right there. So we move our eyes around using those rectus muscles, remember those, to make the image land here. Not here. If the image lands right here, you can't see it at all. In fact, in your textbook, you have a blind spot test. I suggest that you take it. It kind of freaks you out a little bit to learn. There's a spot where if an object is right in front of your face, you cannot see it. It's invisible. That means it's landing right here. If you see it in the greatest detail, that means it's landing right here. So, optic disc on the other side of the optic nerve, macula lutea, and fovea centralis. If I look at this other model internally, it's got a pretty good optic disc right here. In fact, I can see the whole optic nerve shooting out the back of this thing. But where in the world is my macula? This model doesn't have one at all. It's got a nice sclera. It's got a nice choroid, the dark one. It's got a decent retina, but no macula lutea at all. In fact, a lot of models do not show one, but this model right here shows both. Make sure you know the difference of these internal structures in your eye. Now, I think I have hit every other internal structure, and we'll go through a bunch of them in the practice section as well. But for now, let's you and I move our way into the next section, which will be our eyeball dissection.
Yeah, here's looking at you, kid.
And who can't do a little Humphrey Bogart when you get a chance? What we see here are two sheep eyes. I'll dissect one, maybe both of them out for you, and we'll look at those structures that are listed on page 3 at the bottom in your lab manual. Always, my recommendation is do the models first, then the actual dissection. Few things we can see right off the bat with this sheep eye right here. Here is the cornea. Notice it's very cloudy looking. This is very typical. Right after death, it sort of clouds up. Hard to keep the fluids and the cornea clear once this person or sheep has expired. I can also see some little pieces of what look to be almost raw meat on here. These are some of the extrinsic eye muscles, which I do not need for the dissection. So I can cut those off so they're out of our way. Now, sheep eyeball dissection is always, in my opinion, a very good dissection. A little adipose here, packaging the eyeballs as well. And I'll try to keep it in the frame. One thing I always tell people is great about a sheep eyeball dissection. I want you to look at the bulging out of the cornea here. There's a lot of intraocular pressure. And I always tell people when I teach them to dissect an eyeball that this is a great test of your lab partner. Are they a good lab partner or not? If they're a good lab partner, they'll stand right next to me when I make this first incision, because that's when I am most likely to get squirted with eyeball juice. Yes, I did just say that, eyeball juice. By that I really mean aqueous humor. And I have done enough eyeball dissections that I have lost more than one shirt to being squirted with eyeball juice. And if there's a little choroid or other material in there, can stain a light colored shirt, and I just had to throw it away. So this first incision, if my lab partner stands right there with me, accepts the risk, that's a good partner. I want to keep them for the second semester. If my lab partner runs away, hides under the table, ducks behind someone else, and I have seen all of those behaviors, I don't want that lab partner anymore. So you people be good lab partners. So virtually, I want you to stand here right with me, and I'm going to make an incision right here where the cornea becomes the sclera. Remember, these two things are continuous. So I'm going to insert my scalpel right there, and some aqueous humor just shot out of this eyeball. Some aqueous humor, I don't know if you can see it coming out there, that thin watery fluid, that's aqueous humor. And the first thing I want to do, it's a little tough because I don't have a good angle here. I have the camera where I would normally be. The first thing I want to do is cut all the way around this cornea so it can be removed. Now the cornea and the sclera as well, so much collagen, these things are kind of hard to cut. It can be done, you just have to be careful. And keep going around the cornea. And there are other methods for dissecting eyeballs. This just happens to be the Duane method, we'll call it. So you can see the cornea now completely off the eyeball. There it is, I'll just set it right over there. Cloudy cornea. As I look at this thing right now, and I will zoom in a little bit for us, I can see the iris right there, the colored part, and the pupil is this big hole, which is now completely relaxed. Iris right here, pupil right there. I think pretty easy to identify in this eyeball. The next step in my dissection is that I would like this eyeball to be completely in half, two hemispheres. Now that sclera is very tough, but I just put a brand new scalpel blade in here. So I'll do my best to make a good steady cut through the entire sclera. I'll rotate. So I want this thing to be completely in half is what I'm after. And I just managed it right there. So now my eyeball is separated into two perfectly good halves. And I would like you to watch this thing that I'm going to pluck out right here. That is the lens right here. This is the lens of the eye. It's almost spherical looking, isn't it? Now this lens is completely relaxed. It's clouded up. It's sitting right here in some aqueous humor, that thin watery fluid. And what did I pull it out of? This big glop of gel, which is the vitreous humor, which normally, if I can get it to, sort of flops out of the eyeball as a big glob. And in my case, two big globs. There we go. Vitreous humor right here. Now, notice how cloudy it looks. Clear while alive. Lens right here at the end of my forceps. Vitreous humor, and this thin looks like water here. That's the aqueous humor that you can see. I've already seen the iris and the pupil. And what I can see right here very nicely are three things. I want everybody to look closely. Watch very closely what I do with my forceps. See this thing that I'm pulling on right here, this sort of tan or brown structure? This is the retina, the nervous part or the nervous tunic of the eye. Look at how thin it is and how easily it detaches from the rest of the eye. Once the vitreous humor has flopped out, there's not really anything holding it against the posterior wall of the eyeball. And if I work this part of the retina out, there's a little more vitreous humor flopping out. I want you to notice something else right here. It's tough to see in two dimensions. I understand that. But as I pull on this retina, do you notice how it's still attached at one spot right there at the end of my forceps? Let me use a probe to show you. So see how if I move the retina around, it's still attached at this one location right there. That is the blind spot or the optic disc. Let me try to do as close up as I can get. So all this tan, mushy material, so think back to our study of the brain, does it surprise you that unfixed nervous tissue like this would be very soft and mushy? I don't think so. So this is the retina, and right here where it's all connected still to the eyeball, see that? That is the optic disc. And right behind it shoots off, I think you can see it right here, this round structure, the optic nerve, which is right on the other side of where the optic disc is. I can also see the very dark black material right here. That's the choroid. And a specialized section of that choroid right here. Notice this very iridescent blue-green color right here, this is the tapetum lucidum of the sheep's eye. This provides reflection within the eyeball to allow this animal to see better at night than you and I can. You don't have anywhere close to the tapetum lucidum like this. And some people, when they do an eyeball dissection, are very taken aback by sort of how pretty this color is. It reminds them of, you know, mother of pearl, maybe, something like that. So this beautiful colored area is the tapetum lucidum. All this blackness is the choroid layer. And if I turn this thing around, right here on the back side of the iris, I can see little stripes here. This is what? This is that ciliary body. The ciliary body of the eyeball. So let's look at a few things here. Cornea. Let me turn this. Sclera, the white of the eye. Tapetum lucidum. Choroid. That's the retina. On the back of the iris, ciliary body. I can still see a little bit here of my iris and my pupil, which was cut in half. Vitreous humor. Look at how thick it is. Aqueous humor. Look at how thin it is. Well, this other half of the eyeball still has a little bit of vitreous humor, and I can still see that other side of the retina. So see that retina in there? I move the vitreous humor around. See the retina up against the back of the eyeball's wall. One last thing I'll show you. If I scrape away the choroid, what do I see now? That's the sclera, isn't it? So all these parts of the sheep eyeball that I'm allowed to ask you are the ones listed on page 3 in your lab manual. If it's not listed there, I don't get to ask it, everyone. So watch this several times, watch the practice video, make sure that you can see all of these parts in a view very much like this one to get ready for your quiz and also, of course, your practical. Okay, everyone, what you can see here is a very carefully crafted piece of artwork for your viewing pleasure before we look at the histology of the eye, which is really just the histology of the retina, the nervous part back here. So I've sketched out roughly an eyeball. I can see the iris right here, pupil. This is the lens. So when you find the lens, you just move back on your slide until you hit the retina, which is the nervous part of the eye. That's where we'll focus our attention. So I've drawn an extra super thick retina right here, showing us the 1, 2, 3 layers of neurons that we find here. And I drew this out a little bit. You can refer back to this when you talk about it in the lecture, because it gets very confusing sometimes for people who learn this the first time, because visual physiology is really in many ways completely 180 degrees backwards. So what you see right here, the rearmost layer of neurons, these are the photoreceptors, the rods and the cones. Notice I've drawn red, green, and blue cones, black rods here. The photoreceptors actually pointing in the wrong direction away from the incoming light, and we see them synapsing with what I drew in orange, this layer of bipolar neurons, followed by, on your far left, that green layer of ganglion cells. And notice what I did with these axons. They all come off the surface of the retina and sweep in a big cable right back out of the eyeball. This, of course, would be the optic nerve, collecting millions of axons taking them back toward that optic chiasma. See, everything ties together, we hope. So, very dark in the back, I have the pigmented layer, then the rods and the cones, the photoreceptors, the bipolar cells, and the ganglion cells for the histology of the retina. Okay, trying to use this micrograph to give you just a little sense of scale. This is the lower or inferior part of the iris. This would be the pupil right here, and this big thing is the lens. Not that you have to know those for your quiz or your practical. I just want you to see where we are in this eye. And then if I simply go backwards, this is all lens. All the way through this is where vitreous humor would be, everyone, behind the lens. And then I keep going, keep going, keep going, until I finally get here to the retina. So this is the retina. Very easy, pretty straightforward, I think. So what can I ask you on this lab quiz or your practical? What is this a slide of? The retina, the whole thing. Which is really these 3 layers of neurons. And fortunately for us, the nuclei in the neurons takes the most stain, so we can see them quite nicely as 1, 2, 3 layers. Notice the choroid and pigmented layer right here. So all I can ask you back here is pigmented layer. Then we have the rods and the cones, the bipolar cells, and the ganglion cells. So working our way from your right to your left, what do we have? Pigmented layer, rods and cones, the actual photoreceptors, bipolar cells, ganglion cells. This would all be vitreous humor, and see this lighter staining area? These are those axons all bundling up and running out of the optic disc or into the optic foramen. This back here would all be sclera, by the way. Okay, people, listen up and let's go through this ear model. Ha. Now, when we look at the ear, I've probably got, I don't know, I guess if I was to count, my cabinet's open back there, probably about 7 or 8 ear models in this lab room, but they're all typically copies of one another. It's just different versions of this one right here, or maybe the other side, the right ear, but same models, they all look essentially the same. We'll go through this one first and then take a look at a few others as we work our way through. So follow along in your lab manual. This happens to be page 2. And let's talk about the distance from here to here in the ear. What we're looking at here is called the external ear. Now if I rotate this around and angle it a little bit, you might be able to see right in there, right here, a structure called the tympanic membrane. And that's the wall, the boundary between what's known as the external ear and the internal ear and the middle ear. Actually, this is the boundary between external and middle. So the external ear starts out here with this big piece of elastic cartilage covered with skin we have called the auricle or the pinna. Sometimes you might hear that referred to. This is a structure essentially unique to us mammals. We have these. Lots of other organisms have ears, but they can't wear earrings. Sometimes, they might have a tuft of feathers out here or something like that. We actually have elastic cartilage covered with skin. This thing that you clean with a Q-tip, not the ear canal, just this thing, that's the auricle or the pinna. This tunnel that I'm waving my probe in right here is the external auditory canal. And these two things together, the auricle and the external auditory canal, that's the external ear. And that stops right here when I hit the tympanic membrane. The tympanic membrane serves as the wall between what we call the external ear, which would be over here, and the middle ear right here. Now, these two areas, external ear, middle ear, these are typically air-filled anatomical features. They don't have any fluid in them. Oh, you might have a little, remember what's called ceruminous wax in here, but this is air on the other side of the tympanic membrane, and the middle ear is where we work on converting these sound waves into physical vibrations for the inner ear structures. So this middle ear area right here has a few features in it that are worth noting. First off, we have the auditory ossicles, the smallest bones in the human body. If you look at this little plaque I have of them, look at these bones. The malleus, the incus, and the stapes. 3 tiny little bones. Somebody thought this one looked like a hammer, malleus. Somebody thought this looked like an anvil. If you don't know what an anvil looks like, Google it. And somebody thought this one looked like a stirrup, like from a saddle. And I can see that. The malleus, the incus, and the stapes. These are tiny little bones that we find here in the middle ear. Now this model, it happens to be removable to see 2 of them. I can see the malleus right here, which is the first one, that rests against the tympanic membrane. And this one is the incus right here. Now you need to know these names in order. Malleus, the hammer, next to the tympanic membrane. Incus, the anvil, is the second one. And the stapes I can see right there. So these 3 bones are connected. Whoops, let me move my probe out of the way. Sorry about that. Maybe you can see it a little better. Connected to each other and to the tympanic membrane. So if I put this back together, and you imagine right now the sound of my voice entering your ear canal, your external auditory canal, makes your tympanic membrane vibrate, which makes the malleus vibrate, which makes the incus vibrate, which makes the stapes vibrate. We are communicating that sound wave from the external ear through the middle ear toward this snail shell looking thing, which is part of the inner ear for hearing. These 3 auditory ossicles, they're called together, the bones, malleus, incus, stapes, listed in the proper order in your lab manual, I can see right here in the middle ear. These little tiny bones are connected to each other by little tiny synovial joints, believe it or not, between the malleus and the incus and the stapes. Smallest bones in the human body, smallest synovial joints in the human body. Also, little, articulation trivia for you. And before we leave this relatively short area here called the middle ear, notice this structure right here. Find it in your lab manual. This is called the auditory tube. This structure right here is called the auditory tube. It actually serves to connect the back of the oral cavity and this middle ear so that we can always have equalized pressure on either side of the tympanic membrane so that our tympanic membrane doesn't pop that way or pop this way. Think about when you drive up Mount Charleston. If you've never been up there, I suggest you go. It's a great place to take your relatives and family when they come to town. Drive up to Mount Charleston. It looks a lot different than the valley floor. But as we go up in altitude, the pressure outside our head gets less. And if you're a polite person with your mouth shut in the car, the pressure inside your oral cavity stays high, and as we go up the mountain, what happens? Our eardrum goes, our tympanic membrane, and it can go on the way back down. Some people don't like that. So if you open your mouth, yawn, chew gum, like a middle schooler with your mouth open, you can keep the pressure on either side of this tympanic membrane equalized. So this auditory tube, old name the eustachian tube, right here, very easy structure to identify. And just trivia for you, when we were younger, it was a little more horizontal. So bacteria from our oral cavity could get here and cause those middle ear infections that kids get a lot of. As we age, it gets a little more vertical, a little harder for bacteria to get up there. I'm not saying adults can't have a middle ear infection, it's just a lot less common. Let's look at some of these same structures on a different model, just to get a little different view of it here. So external ear or auricle, external auditory canal, auditory tube, tympanic membrane, right there. Tympanic membrane, sorry for the squeak. I'm going to remove a little piece of my temporal bone here. And then I can see on my tympanic membrane, the first bone is the malleus. Rotate, whoops. I just dropped my tympanic membrane. Did you hear that? Ha ha. But if I take it out and look, I can see the malleus and the incus. Most of these models have a removable middle ear. There's my stapes, which looks like it has a fracture. That we'll call that a student-caused fracture here in lab. So those are the three auditory ossicles. One more look. The only other version of an ear model I have, this one happens to be the other side, but I think you can see the structures just as easily here. Auricle, external auditory canal, very nice tympanic membrane right there. Auditory tube. And I think, or at least I hope, very clear auditory ossicles. The big hammer or mallet is the malleus. This one's the incus. Can't see the stapes. And in this model, the stapes is actually permanent right in there. Can't really see much of it. But it does show me the other two ossicles pretty nicely, if you ask me, which I know you were going to. So, tympanic membrane, right there, malleus, incus. Very nice ear model. Then we get to these bony structures that are actually part of the temporal bone that make up what's called the inner ear. the sensory part of the ear for hearing and balance. Now first, let's look at this thing. Let me pull this out. The snail shell. That is the cochlea. That structure is called the cochlea. This is where we do sensory hearing. Then I can see poking up, arcing, these things which are called, I'm going to keep taking it in and out here, semicircular canals. That's what I see here. Semicircular canals. Now I'm going to drop this down and I'm going to raise up my camera a little bit. I think that will help. Angle it downward a little bit for us. And back off on the magnification or the zoom a tiny bit. Because these semicircular canals, there are 3 of them. Anterior, lateral, and posterior. Notice the auricle here. But also I want you to notice the other name for the lateral canal. It's the horizontal one. So I want you to look at this model and I want you to orient the person's head. They are looking straight at you, and which ear is this one? Righty or lefty? Think about it for a moment. I'll wait. Oh, it's the right one. This is a right ear. This is the lateral side. This is the medial side. Why do I do that? Well, this semicircular canal right here, and let me get even closer and even zoomier for you. This one is not only lateral, it's also horizontal. So always look for that one first. The lateral or horizontal semicircular canal. And remember, our person is looking directly at us, correct? That makes this one back here under my thumb the posterior semicircular canal. And this one that's standing right up is the anterior semicircular canal. So, in order, anterior, lateral, posterior semicircular canal. Let's try this. If I rotate this around, so now the person would be looking away from us, correct? They're looking that way. I have anterior, posterior, and lateral semicircular canals. See that? This white stuff would actually be part of my temporal bone. Let's look at the semicircular canals in the other ear models that are fair game for your quiz and practical also. So this ear is the other side. This one is a left ear, isn't it? But I can still see, everybody, my lateral or horizontal semicircular canal right here. This one is posterior, this one is anterior. Anterior, posterior, lateral semicircular canals, and only half of the cochlea right here. One more view. Semicircular canals, notice the ear to orient me. Anterior, posterior, and what's this one? Lateral semicircular canals. Here is the cochlea. Now, another structure we have, which I don't have very many models that show this well, but I have one that shows it fabulously, and that's this one. An isolated model of the inner ear. I want you to look at this one right here. See the cochlea? Beautiful. See the semicircular canals? Lateral, anterior, posterior, like that. But notice what this model has done. I'm going to rotate it. So I see the bony semicircular canal, but then inside I see in gray what's called the semicircular duct. See those terms listed right above the canals. So I have canals and ducts. The ducts are inside. That's what this gray thing is. So the ducts are named, as are the canals, anterior, lateral, and posterior, but they're inside. Many of these inner ear models only show the canals. I can't see any ducts, can I? But this model, it happens to show me both. And another structure to notice here, it's in your list, but it's not in order. Notice the bulge at the end of the duct. Each duct has one. This is called an ampulla. Think about that when you learn about sensory balance in your lectures. This bulge is an ampulla. You can probably see each semicircular canal has one of those. What's in there? Well, some mechanoreceptors you'll talk about for head movement. Two other things that I can see very nicely on this model. So stick with me, everyone. Semicircular canals, cochlea. There's an area between them right here, called the vestibule. The area between the semicircular canals and ducts and the cochlea. This is called the vestibule. And the vestibule has two chambers in it. These two chambers right here, the vestibular chambers, are the utricle and the saccule. This is their names. Look for those names on the bottom side toward the bottom. The utricle and the saccule. These two structures contain sensory receptors. You'll learn about it in a lecture, hopefully, that give us information about head position and linear acceleration, generally we say. How do we remember the names? The utricle is the upper one. The saccule is the lower one. The whole area between my fingers and my thumb right here, that's the vestibule. It has two chambers in it, utricle, upper, saccule, lower. And then, watch this, going to flip this around. I can see the nerve coming out of, and I say out of because this cranial nerve is completely sensory, isn't it? Called the vestibulocochlear nerve. That's this thing, the vestibulocochlear nerve. Do you see how one branch of this nerve comes from the vestibular branch? The larger branch comes out the back of the cochlea, cochlear branch. Vestibular branch and cochlear branch. If I grab another inner ear model, let me rotate it around. So the entire nerve, vestibulocochlear nerve. See the cochlear branch, vestibular branch, I hope. Let me grab another one. Cochlear branch, vestibular branch, see how they're separate, of the vestibulocochlear nerve. I think, or at least hope, that that makes some sense to us. A couple other structures here that could be asked of you on one of these ear models are the two windows. The two windows. Now look at this big one, isolated model. These are the two windows right here. One of them is called the round window. One of them is called the oval window. Which one do you suppose is which? What's this oval-shaped one, called right here, as opposed to this round one right here? Yes, oval window is the oval-shaped one, round window is the round-shaped one. If I look at another inner ear model, do you notice how I only see the round window here? The oval window is actually covered up by the stapes bone in most of these models. Round window is completely visible. Think about this for your lecture. I don't want to steal your lecture instructor's thunder here, but think about this when you learn about these things in lecture. Let's look at this inner ear. So notice my vestibulocochlear nerve, vestibular branch, cochlear branch, and this one shows me both windows. Round, oval. I think you get it. Let's put that back in there. Now, looking at all these structures, I think I have hit all of them that are fair game for us on the quiz or practical from these ear models. Now, you'll notice I didn't say too much about the physiology of hearing. That's for your lecture. But remember, what we're doing is conducting sound waves from outside our head, tympanic membrane, shaky little bones, to make fluids move around inside that cochlea, the hearing part of the ear. I just bring that up because we're going to see that in the histology section. What we have here is our last slide of the semester, just looking at the cochlea, the part of the ear we use for hearing. Now, at this magnification, I can see lots of tubes in this cochlea. Let me pan around a little bit. You can see a bunch of them. I'm just going to focus in on one. I like the look of that one right there, so let's increase our magnification a little bit. Oh, beauteous, right there. And no, I didn't do any editing. This just happens to be a very nice slide. So I want you to look at this thing very carefully. You can pause me, rewind me, consult with some figures that you have in your textbook and your atlas that are very good. But what I want you to see is 1, 2, 3 chambers. These would be fluid-filled chambers in the cochlea, responsible for hearing. The scala vestibuli, that's this top one. The cochlear duct, the middle one. The scala tympani, the bottom one. Yes, these are Latinized terms. So, scala vestibuli, imagine sort of the letter V right here, perhaps. the cochlear duct right in the center of the two. Cochlea starts with C, so does the word center. And scala tympani, I don't have anything better than your toes are at the bottom, so tympani toes, how's that? Scala vestibuli, cochlear duct, scala tympani. And separating these 1, 2, 3 chambers from one another, I have two clearly visible membranes. Right here is the vestibular membrane separating the paralymph-filled, do that for your lecture, paralymph-filled scala vestibuli from the cochlear duct, which is filled with endolymph. The scala tympani filled with paralymph also, and the membrane I see right here separating these two chambers, cochlear duct and scala tympani, is the basilar membrane. The basilar membrane. But you see one other membrane labeled, don't you, in your lab manual? That's this one right here. The tectorial membrane. I'm going to say that again. The tectorial membrane. Vestibular membrane, tectorial membrane, basilar membrane. Now, I want you to pay particular attention to this tectorial membrane we have right here and the structures below it. These are all called hair cells below the tectorial membrane. That's where your hair cells are. Sometimes this structure here is called the spiral organ, the organ of Corti. This is where we hear. This is where our nervous receptors are, mechanoreceptors. If the fluid in this top chamber, the scala vestibuli, starts moving around, that makes the vestibular membrane start moving around, which makes the fluid endolymph in the cochlear duct start moving around. And here's what I want you to notice. It might help you in your lecture. Look at the tectorial membrane right here. And the basilar membrane is this single solitary layer of cells we see right there. As this fluid starts moving around, the basilar membrane, the floor of this cochlear duct, starts moving around as well. It's thinner than the tectorial membrane is. And as that basilar membrane gets bouncing up and down like the floor in a funhouse, the hair cells will impact and move against this tectorial membrane, thereby causing mechanoreception, allowing us to hear. Maybe that'll help you in your lecture a little bit. Then this moving fluid right here, the vibrational waves will be dampened or released through the round window. Study that in your lecture. For lab, scala vestibuli, cochlear duct, scala tympani. Vestibular membrane, tectorial membrane, basilar membrane, hair cells, the cells here, and the structure, the spiral organ or the organ of Corti. Okay, also in this last lab for us of the semester, we do have to know how to test for a few things. So firstly, we have how is it that you test for color blindness? Probably the way many of you are familiar with is this methodology. These are called Ishihara color plates. And what you have a person do is look at these and read what numbers, if any, they see. For example, on the left-hand side, this one has a number 7 in it. On the right-hand side, this one has a number 45 in it. If you look at these two images, these two Ishihara color plates, and you see a different number or you don't see any numbers at all, you probably are colorblind. There are various types of color blindness. Red-green is pretty common. But Ishihara's color plates, that's one test for color blindness, 29 and 74, by the way. Or, next door, another somewhat common method of testing for color blindness is what we call Holmgren yarns. Holmgren yarns. So you take these and match them up as quickly as you can by color. This is a Holmgren yarn test for color blindness. If a person can't do it, of course there may be a problem with their cones, their photopigments, right? Then you see one of these things. That's a Snellen chart to test your vision. We don't use that. What we want is this one. This is an astigmatism chart. You can see it written right at the top. What is an astigmatism? That's an imperfection typically in the cornea, or it could be the lens, where a person's cornea or the lens is warped or slightly misshapen, so light gets refracted weird in one area. It's not the same as being nearsighted or farsighted. And what we use is an astigmatism chart to test for that. So you have a person with one eye closed, focus their attention on the center of this figure, and then they try to determine if all the lines seem equally blurry or equally clear. If everything's clear except, say, the numbers 2 and 8, that's an indication of an astigmatism. If everything's equally blurry because you're nearsighted or farsighted, or everything's equally clear, you probably don't have an astigmatism. But if one section is blurry and the other's clear, there's a difference, that's an indication of an astigmatism. So remember, an astigmatism, a refractive problem, a imperfection in the cornea or the lens that causes light rays to be bent abnormally, using an astigmatism chart to test for that. And the very last thing we have, I don't want to insult you, is how do we localize a stimulus in somebody's skin? Read through the section in your lab manual, but it's very straightforward actually. So have a person blindfolded or not looking, then you poke them somewhere, remove the pressure, and then they use a Sharpie marker or a pencil and try to mark where you poke them. And you measure how far away they are. Now, typically what would be best is 2 Sharpie markers. So I poke my test subject, say right here, with a red marker, then they use a blue one and see how close they get. But of course, then by the end of that, you have a sleeve of red and blue dots if you're doing that test a bunch of times. That's localizing a stimulus, a cutaneous sense. So color blindness, Ishihara's plates, Holmgren's yarns, astigmatism, know the definition and the astigmatism chart, and how do you localize the stimulus? Pretty straightforward, as my expressive hands say, so long for now. Okay, let's see, we did that, we did that, we did that, did that, and I talked about these 3 things. So you know what that means. That means it's the end. We have come to the end of online instruction. Don't forget you have a practice video that accompanies this, but I don't speak during those. So hopefully this semester we've all done as well as we could in the circumstances. I have tried to do my part by presenting you this information. You have tried to do your part by studying the information and taking your lab quizzes and practicals. So once this is done, take a look at that practice video, then you have a quiz over this lab and your practical. So the quiz over lab number 9 and the practical exam, that's all you have left for lab. So make sure that you spend your time Do the practice videos. Get ready for that practical. Go for the highest possible score you can get on that practical to help your lecture grade this semester. I know it's tough. Hopefully, you had a little bit of fun watching the videos. I know I had a little bit of fun making them for you. But it was a good amount of work for me and for you. So give yourself a little pat on the back, but don't do it until the practical is over. Your second practical covers labs 6, 7, 8, and 9. So make sure that you prep for it. That means about 12 or 13 questions from each lab. The same exact stuff is fair game. So look at those lists of things to know for each one of these labs. That's what I can ask you. If it's on the list, I can ask it on your practical. If it's not on the list, I cannot ask it. As simple as that. So, take care, everybody. Good luck in your second semester. I may see some of you then. You may see me then. Or this may be the last time, sorry, you see me. That's okay. You just keep working and good luck in all your future endeavors.